Okay, welcome everybody. I think we'll get started. I heard there's still a few people eating downstairs on their way up, but hopefully they will arrive soon. Maybe turn down a little bit. Can everybody hear me okay? Okay, great. So first of all, welcome. I know a lot of you, maybe not all of you. I'm Mike Friedlander, the Vice President for Health Sciences at Virginia Tech and the Director of the Fralin Biomedical Research Institute. And it is my pleasure and distinct honor to welcome everybody to the kickoff of this year's 2023-2024 Distinguished Public Lecture Series, sponsored and supported by the gentleman you see in that picture, Mr. Maury Strauss, who has been a consistent and strong benefactor of this program. So we are extremely grateful to Maury. And by the way, a few of us had the pleasure of attending his 99th birthday party with him a few weeks ago. So we're really, really honored to have Maury. And I know you probably can't see all the details there, but that's a little poster showing the upcoming lectures in this program. Generally, we run one a month. We have a little bit of a glitch in the spring where we have to have two in the same month based on schedules, but more or less one a month, always on a Thursday, always at this time, always with a reception beforehand. So I hope many of you will attend some of the following lectures as well as we go forward. And I do want to just put in a plug before I introduce tonight's speaker for the next public lecture coming up in a few weeks, shown here on October 19th. And it'll be Dr. Shepard coming, who's the Georgia Athletic Association Distinguished Professor of Atmospheric Sciences. And Dr. Shepard, we're talking about cloudy with a chance of science, the intersection of wicked problems, perception, communication, and risk. And I'm sure most of you have noticed that climate is a little bit in the news lately. And Dr. Shepard is one of the world's experts and a very distinguished and articulate speaker on the subject and issues that might relate to our health as well. So I hope many of you will come out for that next lecture as well, and for the rest of them, as many as possible. Okay, without any more delay, it is really a distinct honor and pleasure to introduce tonight's speaker, Dr. Montague. Reed, I pulled out a picture from a few years ago. I hope that, okay. He's got that look that Reed has. He's staring right into your eyes, right? Whoa. Anyway, Reed holds a number of titles. He is the Virginia Tech Carilion Vernon Mountcastle Research Professor at the Fralin Biomedical Research Institute. For those of you who don't know who Vernon Mountcastle was Dr. Mountcastle actually hailed from Roanoke, Virginia, Roanoke College, and is one of the most famous neuroscientists in the history of the field in the world. Spent his entire career at Johns Hopkins University and made multiple contributions, and through the kindness of the Mountcastle family, enabled us to have a supported position named for him that Reed holds here. In addition, Reed is the director of the FBRI's Center for Human Neuroimaging Research, and he's a professor of physics at Virginia Tech and also of psychiatry and behavioral medicine in our School of Medicine here. Reed's background, a little bit about it: he did his undergraduate work in mathematics at Auburn University. He did his Ph.D. in physiology and biophysics at the University of Alabama at Birmingham. And then he did postdoctoral training and served as a fellow both at the Rockefeller Institute in New York, and then after that at the Salk Institute in San Diego and La Jolla. He's won all kinds of awards. Not going to list them all, just highlight a couple. He was selected as a Kavli Fellow of the National Academy of Sciences China Frontiers in Science program. He's a member of the MacArthur Foundation Research Network on Law and Neuroscience. He was been invited to give numerous prestigious national and global lectures, including the Nobel Mini Symposium in Stockholm recently at the Karolinska Institute. He was selected by the National Institutes of Health to provide guidance on future planning and initiatives in brain research, including the Brain Initiative, which is a major funded initiative of NIH nationwide, and for the New Innovator and Pioneer Awards. He twice won the Michael DeBakey Excellence in Research Award, and I had the honor of hearing him give his second Michael DeBakey lecture when we were at Baylor College of Medicine a few years ago in Houston. And it's fascinating. Many of you probably know the name Michael DeBakey, famous cardiologist, not a neuroscientist. But I remember when Dr. DeBakey was introducing Reed when he was getting that award, and how deeply he had gone into your work and understood it. And it just sort of shows the respect that Reed's work has engendered even beyond the field. He's amongst the very first faculty to be hired here to the Fralin Biomedical Research Institute, along with a few others that are here, Brooks Casas and Pearl Chu. I don't know if Stephen LeConte is here, but there's quite a group that we stole from Houston, came out, and Reed was right at the vanguard of that. Reed has been early into the field of the biology and role of computation of diffusible signals in the brain, particularly nitric oxide signals, which has been, and I think still an emerging hot area. He's a pioneer in the larger field of computational neuroscience. He truly is a key founder of the field of computational psychiatry, and that applies modern analytical tools and functional brain imaging to the diagnosis of mental health disorders. He was an early leader in the field of neuroeconomics and neurolaw. And if this is starting to sound a little strange to some of you aren't neuroscientists, what's this law thing and economics and psychiatry? It just goes to show you all the areas that Reed's work has touched and where he has been a leader and a recognized pioneer in this field. His work has provided some of the most important insights into the underpinnings of how brains make decisions. He's rattled the neuroscience research community, I guarantee you, more than once, with the development of an approach and understanding the brain at a level of software and operating systems. But he's always carefully aware in all of his work and integrating the machinery of the brain itself. He goes way beyond what most scientists coming from traditional fields like biology and psychology have contributed in terms of their perspectives that have been used for decades. He's a technology innovator. He developed what's called hyperscanning, interactive functional brain imaging between multiple sites, including all over the world, centered here in Roanoke, and also the field I mentioned of computational psychiatry. He's a leader in a field using a technology called optically pumped magnetometry to delve deeply and with incredible resolution into the human brain while people interact with each other. And he's developed tools that we'll hear, I think, something about tonight using machine learning to train up sensors to measure in living human brains very important chemical signals that affect all aspects of our behavior. He developed a prediction error reward hypothesis, fundamental revolutionary understanding of how brains carry out the most important functions through calculating the difference between expected and realized rewards from our behaviors. Last but not least, he was a 1981 Southeastern Conference decathlon champion. Without further ado, I welcome everybody.
Okay. All right. Before I start, let me get my thing here. So thank you. I've known Mike for, I guess, 38 years. I was his graduate student back in Birmingham, Alabama, whatever 2023 minus 38 is. And he also won a track race during those years. The intramural 4x400 meter relay at University of Alabama, Birmingham, which had quite substantial athletes that were sort of residues that didn't quite make it into D1 programs. And we were quite victorious. I think you still have that medal hanging on your wall. Yep. I moved away and then kind of grew my career at the Salk Institute and Baylor College of Medicine in Houston. It's a private school, and then Rice University. My work has I'm going to give you a little summary before I start. Has always lived between what is now called artificial intelligence. I'll tell you a little sociological story about it. Neural networks were not considered artificial intelligence not so many years ago. Now, when you say AI, you mean trained by a neural network. That's what it means. That's how much it's transformed. But between that and biology, because there are neural networks living between your ears, right, that have been evolving for 1 billion years. And there's some interesting lessons that we can gain from that, seeing that some of the techniques that we're now using that make these amazing software systems are also living in neurons sitting in your head. Neurons are the main cell class in your head that we think are involved in thinking. They're not the main cell type in the brain. There's another cell type broadly construed called glial cells, about 90% of the cells in the brain. So we have no idea what they're doing. They're packing or something. So I'd like to thank everybody for coming. I see loads of my friends and neighbors here. I've enjoyed living here for the last decade plus, made a real life here. And so it's nice to see people, in the Shire coming out. I had a friend that used to call this place the Shire. We've loved it here, and it's allowed me and my group to do what you kind of have to do with science when you want to do something new, which is you have to get quiet with problems and you have to be a little off the beaten track, and you have to cultivate something. You have to be patient with it. And I will tell you a digested version of what we do. And then I will have 4 slides where you're going to have to take a deep breath and work with me. I won't show any mathematics. I will show one iconic equation, I will not explain it or say where it's derived from and all, but there's actual work behind what I show you. So I see my family. Where's my family here? Can you raise your hands? Oh, there they are, my family. My family never sees me talk about science, in professional settings. I never stop at home. So this will be a newbie for them. So thanks for having me. And I'm happy to be the first local speaker to do this talk. Okay, is this on? Click. No, not advancing. Okay. Oh, is it off? Oh, yeah. Okay. Turn on the advancing instrument before you try to advance. All right, I'll keep my glasses on. Can it see? I don't need my glasses on. Okay, we're going to start with this because this is how all biologists think about biology, and this is how all neurobiologists think about the brain, which is nothing in biology makes sense except in the light of evolution. This is by Dobzhansky, who was at Columbia University, was an evolutionary biologist. And the amount of support for that statement is immense. And nothing in neurobiology makes sense except in the light of behavior. So one of the ways in which you can organize the complexities of the brain's a really complex device, and we're really early days at understanding how it emits all these fantastic perceptions and behaviors. But you have behaviors and perceptions to help you structure the way you look at what's going on inside of it. It's like we found a device on the beach and we watch what it does, and we can use various things that we think are important about what it does to go in and reorganize the way we think about its parts. Neurobiology takes those parts at every level, from single molecules all the way up to behavior. And an institute like this is built around the idea that the real important problems live at the interfaces of time scales and space scales and molecular complexity in the brain. So all the way from single molecule production and control in the brain up to how do you decide whether to eat the chocolate ice cream or the carrot cake, okay? People are very interested in the way in which your brain makes decisions. Policy people, for example, Mike mentioned climate control. Well, what is that? Is that just a policy or political problem? It's a problem of understanding how it is that people in the composite make choices, how to get them to listen to what you're saying, to not be afraid of you, to make rational choices about how we organize ourselves as a society. One of the reasons my work has ventured into economics and to psychiatry and to various other disciplines and the law is that you're the decision maker at the center of all that. People are. And so these are natural excursions of neuroscience into domains that matter. And frankly, it's a sign that it's starting to grow up a little bit. None of that's going to be easy. It's not going to be straightforward, but neuroscience has a lot to say to policy and the way we think about ourselves. Okay, we're going to start with what is a neural network and what is learning. So let's start with what is learning. Learning is this. You have some input, it comes into a box. The box has some sort of internal structure. It emits some measurable output. You have a desired output that you're thinking of there. You compare the two, and you form some sort of criticism. Okay? This can all be mathematized, but this is exactly what it means. Input, here's the output I got. There's the output I want to have. I'm going to use that difference, and I'm going to make a criticism. I'm going to come back into the box, and I'm going to reorganize its structure based on that criticism. That's learning. Okay? This could be a bug. This could be a machine. This could be a program. This could be anything. But that's basically a learning cycle there. Now, the question is, what is it that you're learning, and is it useful to you? This is the way people like me frame the learning problem. What we're very interested in is, how does this reorganization occur? In the brain, that's called plasticity, broadly, and I'll address it a little more specifically later. This criticism is, how do you form differences? How do you decide that you had an error? And are there identifiable neural systems in your brain that take those errors, propagate them into the brain, and make your brain change its structure? And the answer is yes. And the algorithms that the processes in your brains that criticize things are using are the same as the algorithms that people are just now using to train all these fantastical neural network systems. And I'll talk about a couple of them in a minute. So that's a convergence that's started in about 2014. So it's a pretty recent convergence. Over the last 9 years, one of the explosions in neural network learning, and I'll tell you what neural network is in just a second, has a convergence with the kinds of algorithms that evolution put in your head a long time ago, probably 300 or 400 million years ago. So not only are the algorithms old, they're apparently durable. The algorithms that are in a bee's head and in an alligator's head and in a crocodile's head, they're more sophisticated than ours, but the basic components of them are the same, which says something about their durability and their utility in the world that we live in. Okay? Here's a neural network, classic drawing of a neural network. The way to think about neural network learning is simple. If you wanted to write a computer program to solve a problem, what would you do? You'd sit down at a terminal, you'd pick some sort of computer language. You're trying to sort of monitor the temperature in this room. And if it's too high, turn on the air conditioning. And if it's too low, you turn on the heat. Take my phone, sorry. Let me turn my phone off. It's not one of you guys calling me, is it? There we go. If you get anything wrong in that computer program, if one line is wrong, or there's a semicolon out of place, or the data coming in is a little bit incomplete, or it's a little bit off kilter, it's not quite the right kind of data or whatnot, it won't work. And we all know that. Anybody that has anything to do, either a user of output of computers, anybody that's even used PowerPoint or written a document knows that, you know, you don't click on the right thing, something blows up. Okay, you can't make one little mistake. It punishes you. It's like a child, you know, just punishes you. Okay, neural networks do something that we do every day. You train them by showing them a bunch of examples, the way you train a kid. Show it a picture. I'm going to train a kid I'm going to train a neural network to recognize cats. So I'm going to show it a bunch of picture of cats. That'll be on the input side. Okay, and I'm going to label it here. This is a cat. Here's another picture. This is not a cat. This is a cat. This is not a cat. Okay, and I'm going to come back and I'm going to ask the network to identify some novel picture it hasn't seen before. So the first thing it can do is generalize. It forms a model of a cat in every possible configuration, and then it can interpolate or generalize between them. Okay? Well, that's convenient because you often don't know what it is about catness that you need to learn. But a program like this, that's shaped like this, and it's composed of two simple things: units, artificial neurons that have states. The simplest state would be to be on or to be off. And they're connected to one another, and those connections have weights on them. They have numbers associated with them. It's those numbers that change when you say, here's a picture over here, that's a cat. Okay? What I won't do is I won't go into exactly the nature of the feedback information, but it's a criticism, just like I showed you in the other slide. After training it, you'd show it this cat. This is our cat, Romeo. Easy, obviously a cat. Network gets it right. And then I show this creature who was obviously alien spawn. That can't possibly be a cat, but it is a cat. I don't know the name of it, but the network was, you know, not so sure. There's types of machine learning. So a machine doing that would be called a machine learning. That would be a neural network, okay? It's a particular brand of machine learning. One thing to remember about machine learning systems is, unlike us, or unlike a bug or a bee or something like that, they don't know how to take care of themselves. You have to support them, you have to feed them, you have to clean up after them, et cetera. They do not go out and find their own food. They do not recharge their own batteries. They're flimsy in that way. They need people to do that. In fact, if aliens came down to the planet and asked, what is it, the main operation of human beings, it would be to support the air conditioning that blows on all the server farms that we're using all the time, producing all this heat. Think about it. Think about that. The amount of energy used to run computation on the planet, 90 some odd % of it is air conditioning for the server room. Computers waste an enormous amount of energy when they compute, modern computers. We haven't built a computer that's like us. I mean, I see friends and colleagues up here. If I went and touched everybody's head, nobody would be hot. Everybody would be sort of warm to the touch. The computers over in our server room over here, I don't know, what do we have, 15,000 cores or something like that. If the air conditioning broke, they would burst into flames within minutes and melt down. Okay, they're wasting all that heat. Creatures do things just like these systems, plus they feed themselves, they collect their own energy, and they never are but warm to the touch. Your brain runs on 23 watts. Okay, your whole body sitting there right now runs on about 85 watts. Back when you had this, think of a 100-watt light bulb. A 100-watt light bulb is dissipating more energy than your entire body did. You can't even spike your energy unless you're a Tour de France cyclist beyond about 300 or 400 watts, and that's momentarily. Okay, so we run on very, very little energy. That's because we evolved. If you and I are a version of a hominid a few million years ago, and you're 1% less efficient than me, we're not talking about your progeny 1,000 generations later, okay? Being efficient was crucial to staying alive. You have to eat less, and so your algorithms in your head are also efficient for the same reasons. Okay, 3 kinds of machine learning. One is supervised learning. We all know what that is. Someone stands up, raps you on the knuckles and tells you to do something, et cetera. And you can actually train neural networks in the same way. You can say, this is the thing I want you to learn. I want you to reorganize your structure until you learn how to produce this thing. It's a very explicit way to produce an answer here. Then there's unsupervised learning. Can anyone not see any numbers in that at all? Is anybody colorblind? Okay, this is a color blindness test here. But the point is it's made out of dots. So for example, this is the number 16 here. Everybody can see that the number 16 is there. Well, that number 16 isn't written there. Nobody wrote 16 there. The dots have this irregular pattern. If I blew it up and put it in front of you, you might not see a 16. It's a composite assessment of the thing, okay? And this is the kind of ways you ask unsupervised learning. You just sort of present it with data, and statistical regularities are absorbed by the networks and learned by the networks and used. We think that is in large part how you tune up to understanding the world around you after you're born. You come with predispositions for how you're going to see the world, and by unsupervised learning, you kind of organize your sensory structures. If I expose you to a bunch of vertical lines early on in development, and that's all you see, and you go through critical periods of development up until some crucial age, and then I put you in an environment where you have to see horizontal lines, you have a really hard time doing it. It's because your brain has reorganized itself under the vertical environment. Okay, that kind of thing goes on. That's in a sense why when you put somebody in jail cells or they're prisoners of war or something like that, and you deprive them of all kinds of sensory experience, all kinds of hell break loose in their brain, because in fact they start reorganizing, they get in these mood changes, et cetera. Even their ability to sense the perceptual world around them changes. The one I'm going to talk about is at the right. It's called reinforcement learning, and it's kind of what you think it is. It's ring the bell, feed the dog, Ivan Pavlov. So you ring a bell, it's a sensory stimulus, it means nothing to a dog. You wait a little while, then you feed a dog. Can you do it again? Ring the bell, feed the dog. Ring the bell, wait a little while, and all of a sudden the dog's physiological systems have adapted to both the timing and the expectation of what's about to happen. It starts salivating. Its gut starts running. It starts moving. Not only has its brain done that, the neural control of its gut and its salivary glands and all this kind of stuff has adapted to the bell being a proxy for, oh, you're about to get food. We know that. You can do those experiments. If you switch the order, it doesn't happen at all. Feed the dog, wait a little bit, ring the bell. Feed the dog, wait a little bit, ring the bell. Now you come back on Tuesday and you ring the bell. Nothing. Nothing. So there's causality built into the way your nervous system already is absorbing information. Only stuff that follows the predictor is going to cause the system to learn in an appropriate way. That kind of structure, that absorption of causality in the world around us goes on in every creature that moves. So a reinforcement learning system has an agent here. This is the abstract, obviously. And inside of it, it has algorithms for reorganizing its structure, just like I indicated earlier. It also has a policy. And the policy is, given the state I'm in now, what action do I take next? And we'll go back to that in a second, okay? It emits an action. With that action, the environment feeds back 2 kinds of information. It either rewards it or punishes it. And it says you're in a new state. So if I step this way, and I don't get killed, I go, well, maybe I'll take another step forward. Oh, and maybe I get fed, and now I step forward again, and I get shocked. Now the probability of me stepping forward has gone down a bit. You learn this way, and there are identifiable neurons in your brain that absorb that information and use it in a sophisticated way. I'm going to show you evidence of that. The pioneer, I call them revolutionaries. It's kind of an inside joke. They're extremely mild. I know both of these guys. They're mild-mannered people that are, academics in the standard sense of what we mean by that. Poorly clothed and soft-spoken. This is Arthur Samuels. I believe he died the year I got to, he died in 1990. He was writing computer code until he was 88. And he invented the term machine learning. And what he did, he worked for IBM until up toward the end of his career. He wrote programs that learn from experience to play checkers. His algorithm that he used to train that system to learn to play checkers. So you have a checkered board. From any position on the board, there are allowable moves that you can have. And the way you compute in Samuel's mind what to do next is you value the moves. Okay, that's called a valuation function. We all have those in our heads, right? I mean, we do something and we value what we're going to do next, and we typically pick the thing of highest value. Sometimes we pick the thing of not quite the highest value, realize the thing I could have chosen, the counterfactual, was better, back up and do this other thing again. So in a way, his idea of a value function was very modern, and so it learned to play checkers quite well. It wasn't quite in the form of the algorithms that are used now. So that took Rich Sutton writing a thesis at Stanford in 1984 called The Method of Temporal Differences. The instinct behind the method of temporal differences is this. Suppose you're a weather prediction system and you're going to predict the weather. So I predict it's going to, what is today? Thursday? I predict on Monday it's going to rain 5 inches. Okay, it's not Monday yet. Now we take a step forward. Now it's Friday, and I predict Monday it's going to rain 7 inches. So my prediction of 5 inches on Monday here is a little bit different than my prediction of 7 inches. So my expectations for Monday are changing. And he said, those ought to be consistent as possible. It should be that your expectations shouldn't float around like that. Otherwise, it's a bad guess. So one way to critique a good guess is it doesn't, based on new data, dramatically change day to day. Okay? And to that, you add the actual outcome. So you say, I'm going to change my expectations from one day to the next, and I'm going to look at the actual outcome. Then I'm going to change my expectations. Well, that algorithm lives in nerve cells inside your brainstem, and I'll show you a picture of them. I'm pretty confident about that, actually. Versions of that algorithm have differentiated in human beings to have all kinds of versions of it, but that algorithm lives in your brainstem in dopamine neurons. And if you don't think dopamine neurons are important, let me tell you what happens if I take away dopamine neurons. You have Parkinson's disease. You freeze, you can't move. You have depressed mood. At some point, life becomes not worth living. It's a very serious disorder. And they give you drugs to replace, everybody knows somebody with Parkinson's disease. They give you drugs to replace dopamine in your nervous system, and it works for a bit. And then there are an enormous number of downstream consequences from taking these drugs. Many of the what are called dyskinesias are related to taking the drugs to fix Parkinson's disease. Here in Fair Roanoke, we've invented a methodology, here in Roanoke, a methodology for recording dopamine chemistry in the brain in real time and perceiving human beings, and that's the first time that's been done. And we're doing it with Carilion, with Mark Witcher, neurosurgeon partner of ours for, I guess, 13 years. Actually more if we count the time he was at Wake Forest as a resident. And we've developed, through work with Mark, a way to measure this in a human being, both intraoperatively and in something called an epilepsy monitoring unit. I'll come back to that again later. Here are the pioneers of reinforcement learning. This is Andy Bartow, who's also a revolutionary but soft-spoken. The funny thing about the methods, if I were to start writing the equations down, is you'd say, well, gosh, that's very straightforward. That's right, it's straightforward. Its depths have not been plumbed yet in terms of what it can explain and why it is that it's stabilized in our nervous system through phyla, through different kinds of species. These guys are the now called the godfathers of neural networks, Yoshio Bengio, Jeff Hinton, and Jan LeCun. When I was a postdoc, Jan LeCun would come by our lab all the time, and he was working on neural network application. I was a postdoc in a neural network lab. So in science, as you guys may or may not know, you have post-Ph.D. training, just like a medic has post-MD training. They get specialized training in various areas, internal medicine, surgery, et cetera. You in science, you go hang out with people who are doing something you want to do, and you learn extra stuff and extra training. Okay, so I was a newly minted postdoc. I was at the Salk Institute, and Jan LeCun would come by all the time, and he was trying to train networks to recognize handwritten letters. Okay. In 1989, 1990, neural networks were kind of a laughing stock, and it was a backwater. It was called the neural network kind of nuclear winter. That's what we called it. And the reason there was a nuclear winter on neural network learning, which I've shown you here, is because in 1969, a couple of professors at MIT, Marvin Minsky and Seymour Papert, basically said neural networks are no good. They had started in the 1950s with a guy called Frank Rosenblatt and Arthur Samuels and whatnot. And they basically said it can't learn anything. And they produced examples in published papers showing all the things that neural networks couldn't do, and nobody wanted to go into it. And then Terry Sinofsky and Jeffrey Hinton, who you saw in the last slide, teamed up and started making neural network models that were a little bit different and clearly psychologically and biologically poised. So they started saying, well, what if we thought about how you make an inference over shape from shading, and we turn that into a neural network problem? Or how would we train a network to recognize how to pronounce English text? This is something Terry did. So you look at English text, you have the network produce sounds because you have a machine that does that. You compare the sounds to what you want the sounds to be, and you ask, can you train that to read English text and pronounce it correctly? Okay. And so he produced what was a rather cute thing called NetTalk. It was transformative to the field because it just seemed like something really hard to do. We wouldn't say this now, but back then, having something scan a bunch of text and babble away. After a week of training, it was, and you'd hear word fragments and things. And then I took my, and he also, in a moment of genius marketing, he had the output in a child's voice, which was very impactful. And he used to carry the tape around. It was a cassette tape of the network being trained over. It took 2 months to train it back then, right? So, but he would show it at different stages of training, and he had a tape of it, and no one got to touch the tape. He would have a boom box, and he would walk out, and he'd put his little thing in, and he'd play it for you. And when I arrived at the Salk, he played it for me, and then he went back in his office, and he locked it in the drawer. Anyway, it was impressive. It starts out babbling, it ends up pronouncing English speech. We now know it's what's called a 1 layer percent. It's a trivial neural network now. But what he didn't have was enough computing power. So from 1988, when he did that, to 2018, computing power increased by something like 10 million to 100 million fold per unit cost. There's nothing in history, if you framed on the amount of money it cost and normed on that, that's increased 100 million times in 20 or 30 years. Nothing. I mean, I guess I would liken it to a movable press type when the Gutenberg Bible was published and distributed, and everybody thought the world was going to hell in a handbasket then. So we're still here. Anyway, this is Sinofsky, and this is Jeff Hinton at about 30. They're postdocs. I think they're 76 now. Anyway, they invented a neural network system called a Boltzmann machine. I read that paper in college, and I basically wanted to go to work with Sinofsky. So somewhere during my PhD, I wrote him, and I heard nothing back. And I moved away to New York City, and I actually worked with a guy called Jerry Edelman, who had a Nobel Prize, which is not always a good idea for people. And 2.5 years after writing him, I got an envelope in the mail saying, "Oh, hey, I see you write me. You want a job? Where are you?" I said, "Yeah, I wanted a job. 2.5 years. What world do you live in?" And I pulled up, I mean, a few things intervened, but I pulled up stakes and moved to California and kind of never looked back. I quit medical school. I was an MD PhD student, for any MD PhD students here. And I didn't absolutely hate medical school, but I sort of did. I used to tell my grown children that I had failed out. And when they found out I hadn't failed out, it was I got escalated in their mind. Okay, let me tell you now. Do I have a clock in here?
6:08.
Okay, I started at 5:30. Okay, perfect. Okay, I don't know how many people have not heard of the neural network revolution and that neural networks are playing grandmaster chess and driving cars. And has anybody not heard any of this at all, ever? How many people here would admit it if that were true? I believe you. Okay, so we've all heard of that, right? So let me point out a couple of things. So the work that I did with Terry Sinofsky was to work on systems that do what you see lots of systems doing here. And I'm going to start by talking about Atari games. This is the game called Breakout. You move a cursor on the bottom of the screen, it hits a ball, the ball goes up, it knocks a little brick off, and the game is, if you catch it enough times, you have so many hits, you break out through the top, okay? In 2015, there was a Nature paper published by a group of basically neuroscientists, computer scientists at DeepMind, which is a company owned by Google that's in London, showing that they could train a neural network to do superhuman play on 75% of the Atari games, okay? And it was trained using the same algorithms that are in a bee brain, that are in a person's brain, that are in a rodent's brain, or that are a songbird's brain. Exactly the same, okay? Okay, so what? You have a really great Atari playing game. That's 2015. A couple of years later, they produced AlphaGo Lee. AlphaGo Lee was the same sort of thing. They're playing the game Go. Go is a game of capturing territory. You can see it's played on a 19 by 19 board. There are white rocks and black rocks. The idea is there's some set rules and you want to capture territory. It's immensely popular in the East, and it's, I don't know, not popular at all here. For example, if I were to sit down with an 8-year-old in Korea and play Go, they would wipe me out within moments, okay? It was such a popular game there, they had networks where they had color commentators in Go tournaments talking about the power and the adventurousness of so-and-so's play. Lee Sedol has once again shown himself to be the champion of X, Y, and Z. So just a big game, okay? So they took and they train a neural network to play Go. It's easy to train that. There's rigid rules, and there's a binary assessment of outcome. You either win or you lose, okay? It has an immense state space. The state space of a game is the number of legal moves on average that you have from one board position to the next, okay? Let me give you an example. Chess has what's called a branch factor, which is the number of moves that you can make from any position to the next legal move of about 34, 35. I actually don't know exactly what it is. Does anybody know? No? Anybody a chess player, a real chess player? It's like 34 or something like that. Go has a branch factor, the number of moves you can make from any board position on average to the next of 250. The state space, the number of possible games that you could play in Go are so immense that people don't really even know how to estimate mathematically the complexity of the space. It's just gigantic. Well, they had already beaten the Chinese champion, and we were going after Lee Sedol. I mean, it's kind of predatory, right? I mean, and there is probably a backstory where you ask him how he felt. I mean, he was the world's greatest Go player. It was a big game, and I can't imagine him risking his status as the world's greatest Go player. So I think some kudos should go to Lisa et al. Anyway, they trained a neural network on it, but they were a little insecure about what to do from complicated positions. So they had grandmasters inform them. Hey, here's a board position. We don't know what to do next. What are the good moves? What are bad moves? So the grandmaster came in, okay. Well, they beat him 5-1. The first time they played him, they beat him 5-1. That's a match, okay? And then somebody in the company said, well, you know, I think, but when we look at the way it solved the problem, I think that the advice is getting us in trouble. Why don't we just train it from scratch? Tabula rasa, just flat. They've trained it from scratch. That's called AlphaGo 0. And AlphaGo 0 is 10,000 to 0 against AlphaGo Lee. It's never lost a game. Okay. Then they took AlphaGo 0. This is DeepMind. They took AlphaGo 0 and they crafted it to play chess. Because, you know, it's a strategic game. It has rigid rules of how you move your players. And in fact, it has a smaller branch factor, and it can train a grandmaster in something like 10 hours. So you take it from scratch and you produce a new grandmaster in 10 hours. I mean, it's a player, a chess player that none of us could even come close to beating. Then they took the same approach, collected up a bunch of biophysics people who are interested in protein structure. And they said, what happens if we turn the kind of protein structure game, taking DNA sequences that code for proteins and trying to guess the three-dimensional structure of the protein? At the time, there were about 180,000 solved crystal structures of proteins, and they knew what the proteins were. And so they would have the DNA sequence, and they had a way to then map onto and learn to predict the crystal structure, the exact position of the atoms. Okay, so they extract the network, and you have to know the following. The NIH, the National Institute of Health, has probably spent $100 billion over the last 70 years on the protein folding problem, because the structure of a protein is the function of a protein. It's a pump, or it's an ionic channel, or it's a structural structure. Proteins make you go. It catalyzes a biochemical reaction. It does all the work of the biology, okay? To be able to guess from a DNA sequence how it folds up is a big deal. They spent tons of money on that. The problem with it is it's a problem that grows too fast in the size of the problem. It's called, in mathematics, it's called exponentially hard. In other words, as you go from one state, a little polypeptide, and you want to ask, how does it fold when surrounded by salty water, which is inside your body? You can, and we can constrain it because we know something about the chemistry and the folding of the bending of the molecular bonds, but you can't really get anything but approximations. Well, DeepMind, and the guy that did it, whose name is escaping me now, won the Breakthrough Prize last year for it, showed if you train on solved structures, what you can actually get is a lookup table for atom positions. You can look at the DNA. This is not quite true, but it's close enough to true. You can look at the DNA and you can guess the positions of atoms that section of DNA will. No one would have guessed that. People would have guessed, yeah, you have to produce this stuff and it has to live in a solvent. It suggests that evolution selected for that kind of mapping. Well, they've done that. Now they've made protein predictions on 2 million unknown compounds. Okay, that's amazing to me. So that's where I kind of go, man, that is really quite a step forward, and I never would have guessed it. With a game playing program, with just a game playing program. Okay. But one thing to remember is that, and I told you what this valuation function is. In the case of the game of Go, it would be a function that says, okay, here and you're in this board state here. Here are the 250 possible next states that you could have. You could play which one is more valuable in terms of the probability of winning the game. Okay, and it keeps a table like that, and it just picks the most valuable one each time. When people play against the computer, they sometimes get funny, people that are good at Go, that have this deep intuition about it, they get this funny feeling like it's reading their minds, or that it has found parts of the state space that humans couldn't find. Well, it can be exposed to more games. Basically, it's played the history of Go, right? You can't be exposed to 100 billion games. Those same kinds of networks, when you shine them on lesions on your skin now, are so much better at humans of suggesting things to you about treatments because you can train it on 500 million pictures. I couldn't dump 500 million pictures into your head in 17 lifetimes. If I showed you a picture every second, you don't live long enough. And you'd have to be a person who you can't, and your brain isn't built to do that. So those networks are going to be that kind of thing where you scale the data up and you compute these solutions. It's coming in every domain that you could name. You may even have a domain that you know something about where an approach like that would be good. Okay. Let me show you and end on who are these mysterious teachers inside your head that are like these marvelous neural networks. What do they look like? How do they work? And what have we done here that's novel about looking at their function? Okay. So these are the different species here. These are very different species. This is Apis mellifera. This is the Western honeybee. It evolved from carnivorous wasp 125 to 130 million years ago, which means they straddle the dinosaurs. Okay, it's a pretty old species. The kinds of reinforcement learning systems that are in a bee are also in crocodiles. Crocodiles are 400 million years old. Okay. There's the teachers right there. Okay, let me just draw your attention here. So the teacher of the honeybee for whether or not it likes the nectar it's getting and how to learn the association between the flower that yielded nectar and getting the nectar is this strange looking device here. This is a single neuron. It has a neurotransmitter in it called octopamine, which is a chemical that looks like dopamine. Dopamine is the chemical that you are drinking caffeine to impact. When you drink caffeine, it blocks the reuptake of dopamine in your brain, and that feels good. It increases your motivation. It changes your mood state a little bit. Over 90% of people on the planet drink a caffeinated drink every day of the week, okay? We all implicitly know about dopamine system. Well, this is the bee's dopamine. It's got one hydroxyl group different. The molecule is very similar to dopamine. And its role in the bee's learning is just like the role of dopamine in ours. Single neuron, and it projects throughout the bee brain. This is if we took the bee's head and chopped it like this, that's what you're looking at. This is a single axon, which is a connection from one nerve cell to other nerve cells in a rodent brain. Same sort of thing. It's the dopamine axon. The blue and the red are just to highlight where it's making connections. So these are single neurons in small numbers in your brainstem that project to vast tracts of neural tissue, and through rules that we only poorly understand at the terminals, release neurotransmitters. In this case, dopamine, in this case, octopamine, and they're involved in learning. In songbirds, this is a zebrafinch, same sort of arrangement, and they're involved in finding mates, responding to songs, criticizing their own performance. So the way a bird learns to sing is by listening to a male sing to it when it's young. It lays down some templates in its nervous system. As this is a desired output, it compares them, it produces dopamine signals throughout various areas of the brain. It teaches. You have teachers inside your brain that are subject to disease processes. Parkinson's is one of them. Essential tremors is another. Drug addiction is a classic one. Okay? These are old algorithms. Let me just tell you one thing about neurons. So this is a neuron. This is a pyramidal cell from the cerebral cortex of a human. This is the cell body, which means this is what, if you stripped all these things off, there's a nucleus in there and there's DNA in there. These processes are called dendrites. They look like this. They're kind of knobby. It's the thing that these receive connections from other nerve cells. And the way people think about learning and plasticity in the nervous system, this is the business end of that. You have a projection into this, it makes a connection here, and that connection provides drive to either fire the neuron or shut it down, and that drive is modifiable. Okay, so think of it as a rheostat on that little section of the neuron. This neuron takes in about 10,000 connections, so it's about a 10,000 to 1 ratio. There are about 80 to 100 billion of these in your brain, and mainly what your brain is composed of, as you can see, are synapses. This is in a cubic millimeter of tissues, a couple billion synapses, and several miles of biological wire. And it all can heal itself and know how to fix itself, et cetera. We don't know how to build a computer like that. It has to be that it's doing something fiendishly efficient because it runs on very little energy at all, unlike our computers. Okay, you needed to hear that so that you see what it is that these neurons are doing. So there's a psychologist called Donald Hebb at the mid part of the 20th century. He was the first one to really propose the following. When you go around the world, what you notice if you're a psychologist, you're looking at learning, is you tend to absorb associations in the world, and they tend to be present in your nervous system later. Somebody says this word propinquity means X. I heard somebody use propinquity the other day in public, and I remembered it. I didn't remember it. I learned it because I wanted to use it in public, which I've now done. So propinquitous of me. How propinquitous of me. So propinquity, I'd never heard that before. I looked it up. Okay, it went in my head, I've stored it. Okay. Now I can repeat it to you. Well, how did I do that? Synapses, at the very minimum, at least synapses changed in my head. Other things may also have held on to that. What Hebb said was when one neuron produces electrical impulses, which I'm showing here, that run out these things like wires and make an impact on this one. At the same time, this one's making impulses. They run out this process and that one. Then that connection gets stronger, and when they tend to be uncorrelated, it gets weaker. In other words, correlations in the world that you experience get transported into your head by a physiological process that manipulates that synapse. Okay? He's very famous for that. I always hated that idea. It didn't make any sense to me at all. And here's why. Why on earth would you store stuff that's so consistent from one setting to the next that you're going to experience it over and over again and produce all these correlations? And I'm going to waste neural tissue and energy on. Oh, the correlations are still out there in the world. What do I need that for? I want something that can let me act. I want something to let me do the next thing I'm going to do. And so I never liked this idea. I was not in a majority when I did that. One of the famous people that tested Hebb's hypothesis, Eric Kandel, who won the Nobel Prize in 2000, basically for studying the cellular molecular basis of learning, tested it in what's called an aplysia, which is a sea slug, which is basically a bag of salt water, hermaphroditic, and it kind of wiggles around a little bit. It's got 14 neurons or something. It's a simple creature. The more sophisticated of them release toxins when things get near and whatnot. Anyway, they tested Hebb's hypothesis, and you can see it doesn't work. Doesn't even explain learning in aplysia. And what I left off here is it said, but there was this other overlying terminal that had serotonin in it, and it seemed to have some kind of, but we didn't quite figure it out. So when I moved to the Salk, this is Terry Sinofsky, actually I'm in his 70s. He's aging quite well. This is me and Peter Dan, who worked with him. We're aging less well. This is what he told me. Hey, I want you to make a theory of what diffuse ascending substance is doing. I want you to make a theory of what these things are doing. How in the world do they govern learning? And so we didn't look at data. We just sat and talked about it and made mathematical models of it and looked at consequences of it. And we thought, well, maybe we would change Hebb's rule, but just a little bit, okay? And this is what we did. We said, well, there's this thing called a temporal difference prediction error. And I showed you the pioneers of reinforcement learning from before. That was an algorithm one of them had proposed as being really good. We liked the properties of it. We thought it mapped onto there. And we thought, well, the way to do it is to have a correlational mechanism that takes a prediction error. How is what's going on now different than what you expected to happen, and roll it through time. I'm not going to give you any details of this. We worked out the consequences of that. And then we wrote this ridiculously titled paper. This is the worst title of a paper, of any scientific paper ever written. Using aperiodic reinforcement for directed self-organization during development. Probably some of you have it on your coffee table, right next to a brief history of space and time. I used to write papers like that. They were awful. Anyway, we proposed the details of the algorithm there. There are some equations that whatever. The point was, we thought that the dopamine signal, the octopamine signal in the B, the thing that you lose in Parkinson's disease, the thing that directs you to go after food, sex, salt, and water to keep yourself alive, was governed by an expression that looks roughly like that, okay? I'm not going to talk about it. And I'm not going to talk about how it accounts for data that you can measure. But we then ran into data by a German physiologist called Wolfram Schultz, who was a pioneer in the area, who was putting electrodes down into monkey brains while monkeys were doing perceptual tasks and making decisions. And in a sweep, we could just explain 30 years. We could explain every single figure in every single paper for 30 years. Just boom. Okay. Well, that was great. I was very happy about that. And then the paper was rejected serially for 6 years. 6 years. I wrote that paper 11 times. Some of my co-authors wanted to quit, including me. We were unfairly treated, et cetera, et cetera. You know, this isn't really science. You're just making stuff up. You're guessing, blah, blah, blah. The Nobel Symposium last year was on literally that paper. Okay, but it didn't feel like that at the time. So for you people who think that, you know, it's easy work, it's not easy work. It was a brutal time. I remember it as not a good time. Okay. Okay, now I'm going to show you why these things, why learning as governed by these strange looking neurons, like they're like, you know, if they were diseases, it looks like a disease process in your head. These branched things all through your head. You don't have any conscious control over them, and they're governing your learning, maybe implicitly. Learning is crucial, though, to be in control of your behavior from the outside. So we're going to do an experiment here. Okay, this is a thought experiment. These are the same systems that let you assign meaning to a Krispy Kreme donut sign or a piece of art or paraphernalia associated with irresistible drug use. Okay. Here's the thought experiment. So every night, I'm going to, okay, it's a little weird. Every night, I'm going to sneak into your house and I'm going to inject you surreptitiously with fentanyl or heroin or, you know, maybe I have a panoply of options available. An opiate, okay? You're not going to know it. It's going to be like a mosquito bite. I'm going to sneak out. A day later, I'm going to come back, give you a little hit of heroin. A day later, we're going to do this for a month, okay? At the end of the month, I'm going to stop doing it. Well, what happens to you? Well, there are a lot of doctors in here. They know what happens to you. You start getting, your physiology adapts. I mean, you know, you start getting symptoms. You get vagal, you feel bad, your diaphragm, you know, you're sweating, you're in pain, your digestion changes, etc. Are you addicted to heroin? Okay, let's see. Yes. Come on, guys. Come on. Come on. You think of it? Okay. Does anybody think you're not addicted to heroin? Okay. You are not addicted to heroin. You have the mysterious malady. You have no idea where all this stuff is coming from. You have no external symbol to use to associate or reorganize your behavior around in order to chase your drug. You don't even know because I've secretly injected you. The learning part of this is to associate the changes that are going on internally to the circuits I've been describing with an external proxy. Okay? Well, we know that now. And now we know something about the algorithms that the dopamine neurons are using. And now here in Roanoke and the sites that we are supporting around the world, we're actually able to measure dopamine release in target neural sites in human beings that are conscious and doing perception and reward-dependent tasks. I'm not saying we're right on the edge of a giant breakthrough, but we're translating ourselves into situations where, for the first time, we're able to do this in human beings.
Without further ado, please join me in thanking Dr. Montague for listening.